right. All right. Well, listen, welcome everybody to the Large Pelagics Research Center's sixth or fifth annual public seminar series, and it's our third year in Gloucester. So I really appreciate you all turning out. And this is the kickoff of our 4 seminars this spring season. And I'd like to start by introducing, if I can, our next 3 speakers include some really hot scientists as far as the work that they're doing. I didn't mean that in a sexual sense. Sorry about that. Dr. Vince Saba is a sea turtle and expert and oceanographer who will be talking about climate change and leatherbacks. He's actually next week. Dr. Gareth Larson has worked on many different species. He's based at Woods Hole, and he'll be talking about the importance of krill to the Gulf of Maine ecosystem and fisheries. And Dr. John Hoey, who's an old friend and colleague, will be speaking about cooperative research. And John Hoey has worked in both industry and in federal fishery science, and so he's got some really unique insights on cooperative research and science and life actually in the pelagic area. So do turn out for these speakers. I think they'll be really good, good topics. And I really would like to also note that we're dedicating the 2013 series to the memory of our good friend Mark Gottfried, who was a great supporter of bluefin science. He was an incredibly generous man. He helped my graduate students with their samples. Mark is was one of the world's authority on bluefin tuna. And he also was the instigator of our Tagatini tournament. And so keep an eye out in July this summer. We'll have the Tagatini and Mark Gottfried Memorial Tournament, and that's going to be a lot of fun and a family tournament. So we'll keep him in mind. Now, okay, tonight we promised you the title of our presentation is Wicked Good Tuna Research Sheds New Light on the Population Biology of Bluefin Tuna. Now, you can't blame us for cashing in on wicked tuna. But if some of you have been to our talks before, you know that I'm representing all of my lab, our collaborator, graduate students, a lot of people, and actually here's old Doc Edgerton, who was one of the first scientists sitting out using Hodgkins Cove Marine Station, even back when he was at MIT. And actually, I'm very proud, by the way, that some of these graduate students will soon be doctors. Actually, Jessie Knapp was the first one out recently, and you'll hear more about her research in a little bit, and many others. And Angela and Ben are here tonight, so if you have any really specific questions, you can nab them. Ben's over here. Angela's, there she is, she's in the back. Okay, so those are the cast of characters, among others. So, this is what we really look like in our street clothes. Fishermen know that, anyway, enough said. Fishermen have been our cooperative research partners since the early days, and we basically can't get out to the fish without them. We don't have our own research vessel. And so they play a key role and always have in our research. They've helped us focus on our scientific studies that include tagging, aerial surveys, sonar, studies of the distribution of fish, how many fish there are, and what you're going to hear a lot more about tonight will be basic biology and ecology of bluefin. So tonight I'm going to focus more on some aspects, new findings on the most basic aspects of bluefin biology that will affect, we hope, in some point in the near future, stock assessments. So a little bit more of details tonight rather than general. So it all starts for us in 1993 when stakeholders and fishermen have observations that don't match what are included in either stock assessments or management views. And so we have always been inspired to get to the heart of the discrepancies between what fishermen believe is going on in the system and what's actually out there. And as many of you know that have followed our work, that we started in 1993 when there was the proposal that there are a very few number of giant bluefin left in the Western Atlantic. They were looking at a 50% quota reduction. There was one estimate on the lower end of only 7,000 adult fish left in the Western Atlantic. And of course, as soon as we outfitted spotter planes with cameras and GPSs, lo and behold, we sometimes could count as many as 5,000 fish in the surface of a single school of giants in the Gulf of Maine. So as someone that was new to tuna science, it was a real eye-opener for us and my collaborators. So let's back up and have an idea of who's in charge of bluefin management and science. Many of you know that ICAT is the International Commission for the Conservation of Atlantic Tunas. It's a really awkward name to pronounce. But there's 48 Atlantic members at ICAT. They deal with quota allocation, they deal with international monitoring and reporting, and enforcement on the high seas. Within ICAT, there's a scientific body that's known as the SCRS, Standing Committee on Science and Statistics, Research and Statistics, and the scientific advice provided by participating scientists from each country the standard is the best available. That information is then delivered into the management recommendations. And in the US, we're represented primarily by NOAA scientists in the Southeast Fisheries Science Center, but the stars are there because NGO scientists such as ourselves and others can participate in that process. Basically, you have to show up, become a member of the delegation. But as many people know, almost for every stock under management, that the political process and negotiations also determine the final management measures. And you could see the process from the scientific teams down to the politics. Here's Russell Smith, Jane Lubchenco, and others at the head of the US negotiating table. So it's a very complex process. All right, so Tim Lamb has given us our inspiration here of we hope that we engage people into the science and involve innovation. So for Atlantic bluefin tuna, here's a couple basics for you non-fishermen. They're a long-lived species from 25 to 40 years long. They're very fast-growing. They have the highest metabolic rate of all the bony fishes. They have the largest gill surface area that supports their high metabolic rate. And if you also know anything about tuna, they're really unique because they warm their guts, they warm their eyes, they warm their muscles, and so they can really grow fast, and they can travel from the surface to over 3,000 feet, almost on a regular basis. And if you've been to talks about what bluefin do, you've seen that we can show you this repeated diving of bluefin for almost their entire life. So, and as you probably know, you either are a sushi eater or you know about it. There's an extremely expanding market for sushi, not just in Japan and Korea and China, but throughout the world, in Europe and the US and Canada. So, ICAT stock assessments, since they began, ICAT formed in about, I think, 65, are based on what we all know are catches in relation to the fishing effort. And you see a lot about this in the Gloucester Times, about the groundfish stocks. Fisheries scientists rely on catch per unit effort indices of abundance. And also, they rely on the stock trajectories that include the predictions that are based on biological assumptions. And so there's all kinds of information that go into a stock assessment. But the question that we've asked for a long time, we and basically many scientists are the assumptions about bluefin biology and ecology accurate? So the key questions are the key questions for all species. How many fish are there? Where and when do they spawn? What are their foraging grounds? How do they get from their spawning ground to their foraging ground and back? And stock assessment scientists have to concentrate on how many are there and how many will there be in the future. So this is all pretty basic stuff. These scientific questions haven't changed. The basis for Atlantic bluefin tuna scientific management is known as the two-stock theory, or two-stock framework. Here's the stock management line. That's about 45 degrees west longitude. Sorry if I'm standing in your way. The two separate stocks have spawning grounds indicated in yellow. These are known spawning grounds for the Mediterranean or eastern stock in the Med. And the Gulf of Mexico is known to be the primary spawning ground or under management for the western stock. Now, the assumption is based on conventional tagging that showed that the movements, even in historical times, could be across the equator. They could be out of the Med into the North and South Atlantic, and they even crossed the Atlantic. But the management assumption is that the degree of mixing was very low, just under a few % a year. So that was the justification for managing them separately. So even Frank Mather in the 1960s and 50s was tagging fish in the Bahamas that within 55 days were caught by Norwegian purse seiners. So even back then, they knew that bluefin could cross the ocean in a matter of weeks, but still managed separate stocks. So the two-stock hypothesis is still the basis for management at ICAT. And it also assumed, here you see the illustration, that even with the movements, that bluefin will return to their spawning ground for spawning. Now, I think if you've been to any of our talks or have been paying attention to bluefin science, there's emerging science that does not support all of these assumptions under ICAT management. And I think our lab and our collaborator, and particularly my grad students and our colleagues and even our summer people here, the biological samplers, are contributing to this new understanding of bluefin biology. And as you could see, it's pretty basic stuff: guts, gonads, tissue samples, age, and growth. Now, one of the biggest, I think, challenges for the US managers are that the indices of abundance from the 1990s to the late 2000s in the US commercial fishery, as you all know, have gone down until very recently. And so giants appeared to be decreased in number in our area, and we didn't catch our quota starting in about 2004, I think, until very recently. So the question is raised, is that basically the failure to catch our quota or reduction in the catch per unit effort based on a real decline in the abundance of bluefin tuna? Or are there other things at play? So is it a reduction in the spawning stock biomass of Atlantic bluefin tuna or not? Now, given that we have a wide-ranging and complex migration patterns for bluefin, for example, and other indications such as the increase in the availability of giant bluefin in Canada at the same time as our Gulf of Maine commercial catches were going down, could this be an indication that bluefin were moving into other areas and could be another explanation or part of the explanation for the decline in the numbers here? So an alternative hypothesis that there were fewer bluefin is that bluefin patterns shifted to where there was higher availability of prey. And then many of you know that the issue of how much herring we have is a central issue for bluefin tuna and other species, including whales. So Walt Gollett's PhD work, when we were at UNH, really was using fishermen's logs that include not just their catches, but our aerial survey data, the observations of where they saw fish, and Walt used a variety of information, including fishermen's logs, to document basically the geographic center of bluefin tuna from the period of 1980 through about 2006. So this is latitude here, and notice it's up and down. But the centers of distribution as far as longitude shifted steadily to the east, almost about 175 miles. So the data suggests that the center of distribution was moving east and east. And that's also consistent, as some of you know, when the purse seiners that needed to catch schools of 81 inches and higher basically could no longer catch and support a Gloucester share because they couldn't find the schools. So what Walt's work has shown is that during this, here's the period of high seasonal abundance, we were in the Gulf of Maine, but it looks as though the fish were shifting to the east. Now, what we also looked at, and this is also Walt's work, shows the distribution of, now these blue circles, the reds areas too, show the density of herring from trawl surveys. The red indicates the highest density, so those are the hot spots for, again, this is trawl data. It's hard to see these white circles, but they indicate locations where spotter pilots photographed bluefin schools during the times that these surveys were occurring. And the message is that, no surprise to fishermen, of course, the bluefin schools were photographed where herring abundances were particularly high. These happen to be the years that we conducted the aerial surveys. So we can see a trend of bluefin schools following the herring. But it's not a simple story because the herring in the Gulf of Maine have been rebuilt, but what we do see are changes in the age structure of herring and the quality and condition of the herring that are there. And Walt leads a study showing that it's not just quantity, it's also about quality and size. And the condition of older herring has declined on the same time scale as the decline in condition of bluefin tuna that we were documenting. So this, as many of you know, had a huge impact on the US commercial fleet. At the time, the purse seiners could no longer catch enough to stay in business. harpoon and general categories fell off, and the giant bluefin bite shifted to Canada, where, of course, it's still booming business. So it's starting to tell a consistent story that a lot of the decline in the CPUE in the Gulf of Maine could be related to bluefin moving their distribution to chase the prey. All right, so a very central element of bluefin biology is where do they spawn? Now, I mentioned that there's 2 spawning areas in the eastern Atlantic, which is the Mediterranean. These are the Balearic Islands here. And this is the northern Gulf of Mexico, or the loop current. And so it's always been believed that the age of maturity between eastern and western bluefin tuna is vastly different. And that is, in the Med, they're known to be fully mature between the ages of 3 and 5 years of age. The assumed age of maturity in the West, in the Gulf of Mexico, is 8 to 12 years of age, and more recently it's been proposed that they're not sexually mature until they're 16. This is based on the sizes of catches of bycatch in the yellowfin longline fishery, and not on direct observations of fishery-independent studied fish. So for years, bluefin biologists and physiologists have really struggled with this. How could you have a bluefin tuna that eats the same food and has recently been shown to grow at the same rate and mixes in some of the same areas and have vastly different sexual maturity ages? It doesn't make sense to many. So we really delved into the issue of reproduction, sexual maturity, and what we figure are some of the most basic aspects of life for a fish: sex, food, and nostalgia for the places where they had it good. So, as we said, there is this big, you have a question?
Could you go back one slide?
Sure.
That's DPUL spill. Is there any impact there?
Yeah, there's 2 independent studies that estimated for the known area that the area that covers the known spawning area is about 10 to 20% of the area. So the estimates are that the impact is probably relatively minor. And I can follow up on that later. But that's a good question, and you're going to see more to your question in a minute. So the short summary here: eastern and western spawners under ICAT management assumptions. What we do know is that they share feeding grounds, they have same growth curves, and they have similar migration distances to their feeding grounds. So does the western stock really mature at a much later age and larger size? And maybe we should look at the gonads to find out if that's the case. So I just told you that these assumptions at ICAT, that they're very different, but the fish grow at the same rate. So think about it. That would be as if humans grew at the same rate, but had puberty at age 12 versus age 25. So we used things, the basics in, let's call it, this is what shall I say? These are bluefin gonads. What you're looking at are, these are what gonads look like. That's a male, that's a testis. It's a triangular shaped organ. And the females have ovaries that are more round. But this is what they looked like in the foraging ground. And this big piece of flesh here is actually a fat pad. And when tuna come up here, they have very little fat on their gonad. But over the season, by October, if you're a fisherman, you know that that fat pad can weigh 10 or 15 pounds and vastly outsize the gonad itself. And we know from work done in the Med that that fat eventually is used to produce the eggs and the milt, and is used within a period of weeks. The energy, the lipids are transferred to what we call gametes, or eggs and sperm. So that's the energy store. This is what the gonads look like on the Gulf of Mexico spawning ground. There's the male, and there's the ovaries. Those gonads can weigh up to 25 pounds each. So the scale there isn't that good, but these are taken by Jesse Knapp, who looked at basically spawning characteristics in the Gulf. And so the scales are, these are vastly, vastly larger than what the gonads look like on the foraging ground. So what does that tell us? Well, when you look in the Gulf of Mexico or in the Med, you're going to find spawning fish in spawning condition. And we pretty much know that. But you can only examine what you're catching. So you don't know what else is there. And you also don't know where other spawning grounds could be if you're not looking for them. So what we tried to do is focus on the foraging grounds and look at the reproductive characteristics of fish over a much broader area in space and time. And so the big challenge there is because the gonads aren't ripe, you have to use other kinds of measures to figure out if the fish is sexually mature, is it just in a resting state because it's not in spawning, and so on. So it's not easy to do. So what we did is, of course, some of you know fishermen, you've taken me out and our team, my graduate students, and basically we're on the boat. That's Gilad Heinisch here. I think he's on the Karen Lynn. Many of our dealers have helped us, many fishermen. Here's that's Nuno Fregoso weighing a bag of gonads. And here, that's a very small, that's an immature fish, and that's Gilad, very happy, has his gonad. Gilad is a reproductive endocrinologist who's finishing his PhD, hopefully in a couple weeks. Now, when I said that the gonads themselves, when they're not in spawning condition, are really hard to tell what's going on. But thankfully, we have another technique. And just like a human, we can look at hormones. The endocrine hormones tell you what your reproductive status is and where you are in your reproductive cycle. But again, this is not so easy. And this is probably why it was never done before. Oops, sorry. You have to get the brain out. So there, this is Gilad. The brain is in the inside of the head. And Nat and other bluefin fishermen know here what a pain it is to get the otoliths, the ear bones out, and to get, oh, I'm sorry, I keep doing that. The brain, there it is. That's the cavity, and Gilad's pulling out the brain and the pituitary gland. And it has to go from there to liquid nitrogen to basically get the hormones. So we're looking at luteinizing hormone, follicle stimulating hormone, and other aspects of ketotestosterone and estradiol. So that's what we're using. Don't worry, I'm not going to bore you with the details of the reproductive hormones. But I hope you can at least have a couple things in mind. Remember we talked about the age of maturity. In ICAT, the minimum age is 73 inches, and they want to raise it up to much higher in some proposals. So that translates to roughly 73 inches, I'm sorry, this is in metric, 185 centimeters. And what you see here are the reproductive hormones, or a ratio that we use to determine reproductive status or maturity. You're looking at what the ratios are for adult fish that we know are sexually mature. This ratio is very low. But immature fish and young of the year fish over here, that ratio is much higher. So that the follicle stimulating hormone is much more abundant in the brain than luteinizing hormone. And it's absolutely a reliable indicator of maturity. So what did we find for the smaller fish that we looked at? Fish between 134 and 185 centimeters, basically under 46 inches to 73 inches, the hormone profiles look very similar to the known sexually mature fish. And in fact, we determined from a variety of techniques that these younger fish are sexually mature. So this information essentially confirms now the age and size, or the range of age and size of maturity. It doesn't mean that every fish at 134 centimeters is sexually mature, but certainly it's beginning to show us that the western and eastern fish, they have, not only do they have similar growth patterns and they eat the same thing, but they also have similar reproductive profiles. And think about it, this is a big thing, because that means that more fish are contributing to the population. These fish have not been included in previous stock assessments. So this is how new information is likely to affect population estimates once the stock assessment scientists figure out the best ways to use it. And we can basically tell you what percentage of each size class is sexually mature. That's called, it's called a maturity O guide. And we don't have it all done yet, but at least we made that finding. We found that the fish sampled in the Gulf of Maine in the mid-Atlantic, for the most part, above 53 inches, were capable of spawning. It doesn't mean that they spawned, but they were capable of doing it. And studies in other species are that when you first become sexually mature, you might skip a spawning period. You basically don't have it together. But as fish, generally by the second year, most sexually mature fish do spawn. So we do have gaps in sampling between what we call the young of the year, basically the 1-year-olds, and the 42 inches, and we have to fill those gaps. So our results from 2 PhD projects and work with our collaborators in the med do not support the delayed maturity management assumption for Western bluefin. Now, once we determined that all of these younger or smaller fish that we've looked at are sexually mature, then it begs the question, where are they spawning? Are they spawning? Remember that the Gulf of Mexico, which for the most part looks like it only has big fish in it, that's been the basis for the sexual maturity ogive. It's that, well, we only find big fish in the Gulf of Mexico. That means they have to be this big and old to spawn. The current stock assessments assume that spawning is occurring in the Florida Straits and the Gulf of Mexico, but the old guys had it right. Frank Mather, I read a quote from one of his scientific papers. He says, the indications are that bluefin spawn over wide areas and a considerable period of time in the western Atlantic. The giants evidently spawn in the southern areas in April and May, while the mediums apparently spawn north of the Gulf Stream somewhat later. Now, this is hard to believe. I mean, I know fishermen said they found ripe gonads up here, but it seemed pretty far-fetched until we started looking. And we think that Frank had it right. Ben, you can see Ben, Tim Lamb, and myself finally put a paper together that we presented, that Ben presented at the stock assessment in Madrid last September. And we're basically predicting potential Atlantic spawning areas based on our reproductive maturity work and also where satellite tagged fish went. And many of you have heard the previous talks where and you're going to see it in a second, where bluefin tuna go. So we used, I'll just give you a couple details. Oops, we got caught. These are the pop-up satellite tags. They're data loggers. Here you could see with our fishermen partners, we anchored them into the base of the second dorsal fin. That's actually a tag that Mark Genovese tagged from the White Dove 2 back when we were tagging on the purse seiner. They're about $4,000 a piece. They log depth, sea temperature, and light levels. And from light levels, we use basically celestial navigation, estimate times of sunrise and sunset to geolocate. And actually, that's one of Ben's specialties. So we use these tags to reconstruct the migration paths of the fish for the period of time that the tag stayed on. And we asked the tags to stay on for a year. Obviously, they don't always do that, to say the least. But since 1997, we've tagged almost 600 giant bluefin, probably some smaller ones included in there. And this is just, for those of you that haven't seen the data before, the colors don't indicate, these are not just individual tracks, but the colors show you the months of the year. Remember that the management paradigm for western bluefin is that they come up here, they feed in the northern areas where our US and Canadian fleets are accessing them, and the recreational fleet, and they're supposed to all go to the Gulf of Mexico to spawn in April and May before they return. And since the beginning of our tagging, the fish were not obeying the rules. And as you can see, the warm month colors, the April, May, June, sorry, the colors wash out, but the orange, the red, the pink, those are fish located, as you can see, not just in the Gulf of Mexico, but throughout other areas of the Atlantic. And our later results that aren't on this figure show even more diversity of tracks, sometimes going down to the Antilles and elsewhere. This is just, it's a little confusing. Those are med-tagged fish, so just ignore those for now. But the message here is that as soon as we had fishery independent data loggers, we learned that bluefin tuna had much more complex migration paths than had been assumed under management and was consistent with fishermen were guessing that had to be all along. The colors again indicate the time, the month, so they're 12 months. These are 4 individual tracks. The green circle is where the fish was tagged. We tagged these with our fishermen partners in Southwest Nova. That's Fin Seeker. So they're tagged in the same place. These fish were tagged within a couple days of each other, and they're about the same size. I can't remember, it's 500 or 600 pounds. But notice that fish tagged in the same place, the same time, had at least 4 different migration paths over that year, and only one of these 4 fish went to the known spawning ground. So this begged the question, back then, we hadn't done the reproductive work. So it raised the question, are they spawning elsewhere? Are they skip spawning? Well, if you study bluefin and see how fat they get when they're doing well, and the 20 pounds of fat on their gonads, it would be, I think, preposterous to assume that a fish in that condition would forego spawning. Again, it's a hypothesis, but so back in 1997 or 99, our first results were, we found fish that weren't in the Gulf of Mexico, and all we could say is, wow, are they spawning elsewhere? But since then, since 1997, again, these are consistent patterns that we now know the whole annual migration path for many fish. The results are consistent since 1997. Only about a third to a half of our sample each year of giant fish went into the Gulf of Mexico. So this is work led by Ben, and this was a recent publication. And what I want you to focus on is just, these are migration paths of, these are basically giants and fish that would be considered not giants. And remember that these fish are considered, were considered immature. These are patterns by, let's call it quarter. The red dots indicate temperatures above 75 degrees. That red mark is indicating what we consider could be potential larval development areas, because that's the minimum temperature for larval development. So we're saying, okay, we know where these fish went, and the red just indicates sea surface temperatures when fish could be in areas where their larvae could develop, because that's what's important. It's got to be good for the larvae. Bluefin, probably the giants don't care so much. But I want you to notice the extent of the red dots. And they're not just in the summer, but they're even in the winter and the early spring. So it begs the question, could it be that bluefin could be spawning over a much broader time period than has been formerly assumed? There's been independent confirmation that bluefin are spawning outside of the Gulf of Mexico by work done by NOAA colleagues and others. Larval sampling, so these are showing for the first time, when they start looking, their larvae are found outside of the Gulf of Mexico in the Yucatan Straits. And now their models that they're using are predicting larvae in other locations as well. It's not easy to find larvae. You have to have the bongo, you have to tow the right way, in the right place at the right time. It costs a lot of money. But if you want the smoking gun, it's always good to have larvae. All right, so we consider these also the smoking gonads. So the biggest challenge we've had for getting to the gonads is that when you're offshore in the Atlantic, the only people that get out there are the longliners. It's extremely difficult to get large numbers of samples when longliners are only allowed to board 2 fish, and that's only if they have their hold filled with their target species. So since probably the early 2000s, we've been working with longliners to get gonad samples, but it's hard to do. But finally, we got Bart's not here, Bart DiFiore on a longliner, and we kind of know where we want to go, but we have to put somebody on the longliner in order to be permitted to do this work. So here's an example of a smoking gun. Longliners off the east coast of Florida in the Bahamas. It said there's always a run of ripe fish from the Antilles. They're going north. They're not coming from the Gulf of Mexico. I mean, who really knows? But they generally tend to follow the fish. So these are clearly fish. We did the histological work, getting the samples, and they were within 2 days of spawning. That's a ripe female. So, okay, it's only one. We actually had about 4 fish out of a sample of 6. 2 of them were post-spawning, but most of them were very close to spawning. Now, this is the interesting part, tantalizing. These are gonads that are, this is the Eagle Eye, one of our cooperative partners, and those are ovaries that were taken on July 4th on George's Bank. So way, way past the spawning ground and past the known spawning period. So is this a fluke? We've known from people like Mark Gottfried and other fishermen that we've worked with and talked with over the years that they often get gonads in that, or at least some of the time, in what they consider and what we found to be ripe condition. What's interesting is that these are gonads that Jesse sampled in the Gulf of Mexico. They look the same. They are the same. Their gonadal indexes are very similar. So it's very tantalizing. Could this be evidence, or at least very, very small evidence, that Frank Mather was right about spawning in the late spring, early summer on the Gulf Stream margins off New England? I didn't think it would have been possible, but when you get the ovaries and the gonads, it's looking more and more plausible because you've got long day length and you've got warm temperatures in the Gulf Stream. So the conclusion of the reproductive work is that we believe that bluefin tuna are also likely to spawn outside of the Gulf of Mexico and in the Atlantic, and over a much broader space and time than has been assumed so far in the basis for management. And we're also suspecting that smaller fish don't go as far, that there's probably spatial and time distribution based on the size of the fish. But as any good scientist will say, well, yeah, but you haven't proved it. And absolutely comprehensively, and absolutely right, we have not. So the global we is we need more evidence. We need much more extensive sampling over time and space. Probably would be also really good to have larval surveys over broad regions. But we all know funding realities. There's less and less money for scientific cruises, so it's going to be hard to do this. There's less and less money for biological research on bluefin. So just as we're getting to getting it all together, scientists are up against it, that the money is going away. So I just had to say that. So to finish up, too, with a little bit more of the biology, as you've probably seen at other talks, that we've been using these pop-up satellite tags. Now that they're smaller, or mini pop-ups, we can put them on smaller fish. As other people are using them on other species, including squid and jellyfish. But anyway, here we're tagging on the Tammy Rose, and we're getting year-long migration paths for the juveniles. So here's just one example. Here's where we tagged it at the BE. No, can't remember. They moved the BB. BC buoy or the regal sword. We're missing a bit of data, but here's October. Remember, the warm colors are the warm months. And the triangle, you can't see it. That's where the tag came off a year later. And on the left, you see the very fine scale temperature and depth record, and actually temperature at depth over that period of time from October through July. So for each of these tags, we know depth and temperature records for each of these fish that we have tags. So it's a really comprehensive story. So this fish is 48 inches. Now, when we started tagging, there was no way that we considered that any of these fish could be sexually mature. We now know that some of these fish are probably beginning to mature. So the question is, where are these smaller fish going to spawn, or are they going to spawn? So what Ben has shown you here, this is also what we call a habitat utilization. So the blue area contains all of the pop-up tag locations, and the red and the yellow show the highest density of where the data records were. So you can see January, February, March, you can see that the fish have a pretty broad area. This broad area includes the Bahamas region, the Antilles, the east side of the Gulf Stream, where temperatures are possibly warm enough to support larval development for some of these fish if they are in fact sexually maturing. So stay tuned. This is a big part of the story because there's a lot more of the smaller, younger fish. If they're sexually mature and contributing to the spawning stock biomass, that is also going to affect stock assessments, we hope. And in fact, there's beginning in sensitivity runs consider some of this new information. And we hope in the next assessment in 2014 that they'll start using it. Again, another really important part of our program, Emily Chandler, program manager for Large Pelagics, who's in the back, has been running with many fishermen's participation, the Tag a Tiny program. This uses a conventional tag. And there's lots of reasons why we do this, but it's actually been really exciting. This program, which is, let's see if I can get some more up. We did, by the way, that was for Eric Stewart. He's not here tonight, who is with Jeff Corwin. And this is our NOAA grant manager. We did a lot of our satellite tagging for juveniles on the Tammy Rose and some others. But over time, we are following year classes with our data from Tag a Tiny. So not just the landings from the rec fleet, but you can see the young of the year start here. That's the sizes. And as time goes, what you see, I know Nat and Derek know this, in our own data, just from the Tag a Tiny program, we see year classes moving through the fishery and changing each year from these very small young of the year that we generally see off the eastern shore in Maryland. And this is one way to find out who's here. But again, this is the fishery-dependent data. Let me see if I can get this to go. So really a popular program. We're really pleased. We have over 1,600 volunteer tags deployed, over 1,200 volunteer anglers. So check us out if you want to participate. And if you go to our website and Facebook page, you'll see that not only have we gotten fish that fishermen in the Bay of Biscay and the Med have tagged here, caught by our wreck fishermen, but some of the fish tagged by people like Jules Boudreau and others have been recaptured in Morocco, in traps, and more recently in the Balearic Islands on the Med spawning ground. So this story is not simple, and it's a lot more complex than has been assumed. And finally, for stock assessment scientists, how many bluefin are there? Remember I said that ICAT uses primarily catch-based fisheries statistics to estimate population sizes and trends, and that's known as fishery-dependent indices. 10 out of 11 or 11 out of 12 used for the Western Atlantic are fishery-dependent. But since 1993, we've been trying to develop fishery-independent ways of counting fish, starting with aerial surveys with spotter planes. And back then, and I mentioned that, that's how we got into bluefin science. So really, I think the innovative aspect of our work, we were among the first to use the pop-up satellite tags. We are among the first to use sonar and acoustic methods to count fish, and certainly aerial surveys for bluefin tuna. And the goal is to keep using new technologies to find better ways to develop fishery-independent indices of abundance. And so Angela Vanderlaan is leading our efforts to count fish. And believe me, they don't usually look like this. This is spotter pilot Wayne Davis, his company, I forget what he calls his company. He has some of the most beautiful aerial images of marine animals, and he's come a long way since being one of our spotter pilot volunteers. But they're absolutely beautiful from the air. But this is more or less what we have to look at. Here you see, to know what we're up against, we have to have the boat in the frame. This is the Lily. This is Billy Menese's vessel that we've worked with for a number of years. There's Mark Brochu is our spotter pilot. So his job, and Ben's, who's in the plane, is to get the school and the boat in the same frame. That's so we can geo-reference, geolocate, and know exactly, basically calibrate our images for sizes. From these photos, Angela and her team and our sonar collaborators are able to estimate school shape and size, the number and size of the bluefin tuna, their packing density, and so on. This hasn't been done before. And in fact, the EU is now starting to pay attention to our methods, and we met with EU scientists last December trying to develop similar methodologies. So sharing expertise and trying to push the limit, because back in the 90s, spotters could tell us how big these were, but actually NOAA did not want to. Let's just say they were uncomfortable using anecdotal information. So now we're trying to deliver quantifiable, verifiable data. So again, just as I started, the number of people and scientists, fishermen, volunteers participating in all of this work is vast. We're very, very proud. We're a small group out at Hodgkins Cove, but we work with a very top team of international scientists and fishermen. There's Mark Brochu, who's up in the air, and here's Billy Menese and our team on the water, and our European collaborators here, and the University of New Hampshire's Center for Coastal and Ocean Mapping. So I hope from these photographs you also get the idea that bluefin science is international, and it has to be, because bluefin are clearly a fish without borders, and our collaborators come from the Basque Country, from Spain, from France, Israel, who do I live out? Italy, and we're really, really pleased. For a small lab, we've been able to really, I think, make a difference. And so this is where I'm going to end, more or less. I must point out the really strategic and important support for our science from our partners and donors, Cell Signaling and New England Biolabs, and the Guy Harvey Ocean Foundation. They've really stepped up when government funding has fallen off to keep us in the business. And our funders on our grants and contracts, of course, NOAA, and our collaborators there, NOAA scientists. So again, our thanks to all of them. And here, we don't have everybody on here, but I'd like to acknowledge all of our collaborators, our grad students, our undergrad interns, and just about everybody else, and particularly our fishermen partners. So thanks very much, and I'm going to stop here. I think I have time for a couple of questions. Nicola.
You opened the discussion with the ICAT fish assessment in the, I think, early 90s of about 5,000 or 7,000, maybe you said, large tuna. Closer to the end, I was hoping you'd give us a different assessment, if you have one, from your studies or not.
No, that's a really big question. The goal is, between the EU scientists working on direct assessment and our team, which also includes our NOAA partners, we basically have to work out the technology, the sonar and the aerial. And we're doing this on the cheap. I mean, literally supported by a Northeast Consortium grant. Once we show feasibility, we hope that we'll convince the governments that this is worth investing in. And Angela Vanderlaan is particularly interested in designing the surveys. How do you do this? They're not air breathers. They're not marine mammals. They're not turtles. So how do you figure out the cost, basically the most cost-effective way to count the fish and to lay your survey lines? And I do believe that within 3 years we'll have it together. But then you have to ask governments to step up and fund those surveys as they do trawl surveys and other things. And we also think that that it can be done using fishermen partners, that we don't need research vessels. My goal would be, and our goal would be, to outfit the fleet to participate as they have in a much broader way in bluefin science and assessment.
So if the 50-inch class fish are spawning, and you take some conservative estimates, maybe they missed one season, what do you think that does to the biomass assessment? I mean, back of the envelope, what kind of percentage are we underestimating by it?
Well, it's really, I mean, these are early days, and I'll say this right out, that they're just, particularly the females, the males are, everyone we've looked at is mature at that size. The females, it's a range. And I will point out that there is the caveat that we know that some eastern fish come over as juveniles and forage here, and that's from biomarker studies and genetics. So it is true that there's probably some med fish in our sample, but we know that can't be all of them. And then other studies show that they're not all med, that they certainly can be med fish. So getting to your question, Ben Galuardi was at the stock assessment when they did what they call a run. If they change lower the age of maturity, I believe that Ben said that one run was that it could affect spawning stock biomass for the West by, I use the quotes, 20%. I don't know where Ben is, if I got that right. But it would be a modest number. But the much larger effect would be demonstrating that spawning is occurring over a much broader space and time, because right now the estimates are also based on this assumption of only northern Gulf of Mexico loop current. What's interesting is the Mexicans have data that show that bluefin are spawning in other areas of the Gulf of Mexico and at smaller sizes. And we have tag data that show that fish are in the Gulf of Mexico beginning at Thanksgiving. So if you look at the whole picture of the area that bluefin could be spawning in space and time, that's going to have a much bigger impact on, we think, on the stock, one would think, the estimate of biomass. What's also interesting, does it mean that all of a sudden there's so many more bluefin? No, it is what it is. It just means that the estimates might explain why, when the predictions were that the bluefin are on the brink, people that actually study bluefin and did the work out in the field, which I hope we include ourselves, we thought that's outlandish. And in fact, my message on saying, who are the experts are ICAT and bluefin scientists. They're not organizations that have a particular point of view about management policy. There's expertise and expertise. ICAT is the authority on bluefin science, the best available. And that's where we look to the best science. And so we believe that, yes, there's more bluefin out there. Alain Fontenot, who is one of the best, longest participant, French scientist who knows tuna fleets cold, big-eye bluefin, you name it. Alain Fontenot always said, it's the myth of the virtual population assessment that's the basis for bluefin assessment in the Atlantic. He said it's a complete myth, Molly, because, as there's a Frenchman in the thing, because he said if there were as many fish as we said there were, and in the estimate, they would have been extinct in the 1980s. Of course there are many, many more fish. Alain Fontenot was probably absolutely right, because we know that bluefin, we can't count all the bluefin when we're only counting the catches that are held to quotas. So to me as a biologist, I always thought it was absurd that people say, oh, you're undermining conservation. science is science. We count the fish, and if there's more fish out there, that's what we have to deal with. So it doesn't mean that they're not in trouble or that there are not other issues. But it's certainly probably, it looks very, very likely that there are a lot more bluefin than are being counted right now in the current assessments. Good question.
The monster fish, could it be that there's just the same amount, that the biomass will be the same, but just the monster fish are gone and there's a bunch of midgets out there?
Yeah. What you're asking, have we taken out all the big ones? And there's definitely evidence that there are fewer monster fish, as you say. And there's, in some fisheries, they say, well, if you take out all the big ones, you're going to exert, pressure to cause the bluefin to spawn at a younger size and age. And in some species, there is evidence for that. But I would say that there's probably absolutely fewer adult or big old fish. They certainly exist. They're found in the Gulf of St. Lawrence. But there's no evidence that the age of maturity has come down. Bluefin have been fished since before Christ. They're the basis for trade in the Med in AD. And so there's been observations on sizes and maturity for bluefin probably is older than most fish in the ocean. So, but yeah, you're right. Certainly humans have exerted influences on fish, but whether it's made bluefin younger, fishing pressure has made bluefin younger, it doesn't appear to be the case so far. Bart.
What do we know about the Pacific bluefin tuna? And is there anything we can extrapolate there as far as when they spawn?
Well, they're very same body plan, but Pacific bluefin appear to be much more heavily fished, and the management measures were much less than in the Atlantic and southern bluefin. Finally, it's looking like the Pacific Regional Fisheries Management Organization is paying attention, and it's maybe a little too late. But they're definitely, everyone agrees, they're vastly overfished. And in answer to your question, it's known that bluefin in the Pacific spawn in the Western Pacific. And so our Japanese colleagues have tagged juveniles with implanted tags, so they know the migration routes. What's really interesting is they spawn over a vast area in the Pacific, over space and time, just as we're predicting in the east. And then they do like Atlantic bluefin. They cross to the east and forage in the eastern Pacific in the really productive coastal areas off Mexico and Southern California, and then presumably return to the west to spawn. But there's lots to be learned, and the fishing pressure is mostly in the Western Pacific. And then in the Eastern Pacific, of course, with purse seining off of Mexico. So, George.
You mentioned that, of course, the tuna feed on herring, but you said that the quality of the herring has diminished?
Yeah, well, that's their primary food, and it's not their only food. Certainly, herring, mackerel, offshore bluefin almost always have squid. And we really suspect that squid populations could drive, in fact, a lot of what's going on the coast. Nobody knows anything about these squids. That's really sad. But so here, what was going on is Walt's work looked at the condition of herring and bluefin. And over a period of time, if there's dealers here, they know that the condition of bluefin declined. What's interesting is Walt found that the condition of herring also declined at the same time. So even though the stock assessments indicated that there was a high abundance of herring in the Gulf of Maine, that the size classes were smaller and the condition was also lower. So it looked as though there was a clear link between the condition of the prey and the predator. And in fact, in whales and marine mammals and seabirds and the Canadian shelf, there were similar phenomena. So you are what you eat, for sure. The good news is that that trend is reversing, and the dealers and fishermen know that the quality's increased in both the herring and bluefin, and the prices for bluefin have gone up. But definitely in Canada, mackerel, capelin, sand lance for juvenile bluefin, haddock herring, we found all kinds of stuff in stomachs. But herring is what they really like. The fattier the fish, the better. And if herring isn't abundant enough in the good condition, what basically the hypothesis is that the bluefin have followed the good herring out of the Gulf of Maine and to the east and to the north. And I left somebody. Okay, I'll get to you.
You said that the oil spill only was a minor impact on tuna in the Gulf of Mexico. In the settlement with BP, did that provide any resources for additional science?
Wow, you just said, thank you for that. That's a really good point. So the estimates from other studies were, well, there's two. They thought 10 to 20%, and you could see from our data, we're predicting that there's other spawning grounds. So that also, right. But it doesn't mean that the adult fish, or what they're for to the fish that were in the Gulf of Mexico that our fishermen catch, were they affected? We don't know. We're now starting to look at parasite loads, at basically the health of tuna. Now, personally, do I think there's a connection? I have no idea, but we've been noticing an increase in parasites, internal parasites the last 2 years. I have no idea if I'm just paying more attention, but we can't get to the funding because the funding for the Gulf of Mexico tuna, the large pelagic species, is so far as for contiguous states, and so it would be to researchers based in Texas, Florida. But of course, these fish are traveling, and they're the fish that some of them. So we would like to be able to call attention to that bluefin and other species like yellowfin swordfish and others enter the Gulf of Mexico and then leave. But overall, we should pay more attention to the health of these fish anyway. Very few studies have been done on health of large pelagics. I will tell you too, one thing is we've taken all our samples that we've archived over the last 10 years that grad students have collected, and we finally basically took a load of hundreds and hundreds of samples that our biological samplers and Emily Chandler helped organize, and they've been shipped to a leading mercury lab. And we're finally going to hopefully settle the question, what are mercury loads in commercial caught bluefin? Not just one fish, you know, that you see the headlines, oh, there's mercury. What's the load of bluefin in mercury in bluefin? What's the selenium in bluefin? And we hope to have definitive answers on mercury.
So are they looking at things other than shrimp and oysters?
They're looking at.
Would they look at bluefin?
There are colleagues that are at Texas A&M that are, and National Marine Fisheries have received lots of money to put satellite tags on bluefin and yellowfin in the Gulf of Mexico. Yeah. So they're conducting tagging studies of fish there. But we haven't been able to get to the money yet, so hopefully in the future. We'll see. Question?
Correlating alternative ways of collecting the data using something like what happened in Fukushima and using those pathogens that they're seeing in the fish being caught off the west coast of North America. Mm-hmm. Are there alternatives of like providing marker chemicals that you could use much less expensive than a pop-up or something like that are being developed?
Well, I think, yes, they've already, you're right. I mean, I didn't talk about it at this talk, but there are labs that are using biomarkers, namely isotope ratios. So you can tell if you use, say, oxygen and carbon and other what elements, they characterize ocean masses, like the med looks different from the Gulf of Mexico or the Western. So, for example, when I said that we know we've got a lot of eastern fish, between 50 and 70% of the samples of juvenile bluefin that Rebecca Dickhut, who sadly is gone now, and Jay Rooker and Dave Secor and others found that they have signatures of eastern fish. So that's how we know that the mixing rates from the Med and East in the recreational fishery here is really subsidized by the East. So that's a really good point. So that's a good point. We can use, Rebecca Dickhut developed a method to use PCBs that are only allowed in the Med as a marker. But the thing is, as the PCBs decline, as the fish grows, you can only use it for about 3 years. And then, so yeah, absolutely. And just a spaghetti tag tells you where the fish was tagged. But the issue is, as you could see, the pop-up tags generally could keep them on a year, and then, you know, the mono fails, something fails, or the batteries. So we can't track using an external tag for 3 years. The implanted archivals, you could say get 3 years in theory if you caught the fish and got the tag back. So, but what you want to know is, what are the migration, how many times does it cross the Atlantic, and where does it spawn? And we still can't tell you if those so-called eastern fish, the fish that come from the Med, do they go back to spawn? Because if I showed you more of the small fish tracks, we have yet to see a fish go east of Bahamas. And now we know that probably some of those fish that we tagged were sexually mature. Is there spawning site fidelity? Do they all go back to where they spawn? That's the management paradigm. But could it be that spawning is looser, that it could occur at different, say, water mass? We don't know. It makes sense that they go back to the same place, more or less, but if that's going on, when does it happen? So that's a still hole in our understanding, and we'd like to know. But the problem is that just when you have all the questions, now the funding goes away for the electronic tagging, so it'll take a while to get back, I think. But we do hope to figure it out. We want to know when eastern fish go back, or do they stay here and end up spawning in the west? We don't know the answer.